Hello everyone, today we are at the CERIC headquarters in Trieste with Alfredo De Biasio, who will tell us about his research results. Alfredo, can you tell us a bit about your research?
Sure. Our research in the structural biology laboratory at Elettra-Sincrotrone Trieste focuses on the fundamental biological process called DNA replication. So before dividing, cells need to accurately duplicate their DNA to ensure that each daughter cell has an identical copy. The process is carried out by enzymes called polymerases. In order to function, the polymerases require the so-called DNA sliding clamps. There are ring-shaped proteins that encircle DNA and prevent the polymerases from falling off the DNA. So in our laboratory, we study the three-dimensional shape and motions of these proteins, and in particular of the human sliding clamp PCNA.
Okay, and what are the results that you came up to?
So we wanted to resolve a long-standing mystery that surrounds PCNA. How does it slide on DNA? Is its motion controlled by chemical interactions with DNA, or does it rather levitate on DNA? So our work allows for the first time to visualize at an atomic level the interactions between PCNA and DNA and to follow how they evolve in time. This provides the molecular basis for explaining how PCNA slides on DNA, helical motion based on short-lived polar interactions. This also sheds new light on previous biological observations on PCNA function in DNA replication.
Okay, in this process, what are the techniques and facilities that you have used?
So in this study, we used a combination of X-ray crystallography, nuclear magnetic resonance, and computational data to investigate the mechanics of sliding of PCNA on DNA. Our work is collaborative and has been possible thanks to the contribution of the NMR team headed by Francisco Blanco at CERIC in Bilbao, and the computational team headed by Ramon Creuget at the Institute of Advanced Chemistry in Barcelona. We also acknowledge CERIC for the access to the X-ray beamline at Elettra-Sincrotrone.
In your research highlights, you have mentioned that this study is very important for future medical applications. Can you explain to us how?
Yes. So, owing to PCNA essential function in cell proliferation, there has been a significant interest in the role of PCNA in cancer, and PCNA inhibition is considered a valuable anti-cancer strategy. This led to the development of specific small molecules that block PCNA function. All these strategies target the PCNA external surface. So the unveiling of the mechanism of PCNA sliding and the identification of new regulatory mechanisms occurring at the PCNA inner surface offer a novel and unexpected opportunity to develop inhibitory molecules specifically targeting the internal region.
Okay, thank you so much, Alfredo, for taking the time to share with us your research and the results that you have came up to.
Thank you for this opportunity to disseminate our results.